Moving on to the next topic, the controversy of spontaneous generation theory. Scientists began to wonder about the origin of animalcules revealed by Leeuwenhoek. It was supposed that these animalcules originated from non-living things. This was called the theory of spontaneous generation or abiogenesis. It was thought that maggots could arise from rotting meat, mice from corn, flies from bovine manure, and fish from the mud of previously dry lakes, and so on. But Italian physician Francesco Redi doubted this. He set out to prove that decaying meat kept isolated from flies never developed maggots, while meat exposed to flies was found to be covered by maggots. He was a strong opponent of spontaneous generation theory and believed that maggots developed from eggs laid by flies. To test his hypothesis, he set out meat in 3 jars. One open to the air, second sealed completely, and the third covered with gauze. As shown in the figure, the meat inside the unsealed jar became infested by maggots, whereas no maggots developed on the meat kept in the sealed jar. However, in the jar covered with wire gauze, the smell of the meat attracted flies but could not enter the flask. Hence, maggots developed on the wire gauze. These results compelled the scientists to believe that living things could not originate from non-living things and arose from only other living things. This was one of the first examples of an experiment in the modern sense in which controls were used. In spite of his well-executed experiment, the belief in spontaneous generation remained very strong. John Needham was the strongest supporter of spontaneous generation theory. He boiled meat infusions in flasks and sealed them with cork. Some days later, the flasks showed the presence of a number of microorganisms. He explained that these microorganisms had arisen from inanimate infusions since he had heated the flasks to kill everything inside. The Royal Society was so very impressed by Needham's experiments that they elected him a member. Italian naturalist Lazarus Palanzani was a strong opponent of spontaneous generation theory and was one of the first to provide strong evidence that microorganisms do not arise spontaneously from meat or hay infusions. He took 2 flasks containing hay infusions and boiled them for almost an hour. One flask was kept open to serve as the control, and the other was sealed by melting the neck, which he called hermetic sealing. He believed that mechanical sealing with corks was not satisfactory. His infusions in the sealed flask remained free from microorganisms for a long period, whereas microbial growth could be seen in the open flask. He concluded that Needham had probably failed to heat his flasks sufficiently to kill the microorganisms. The microbes exist in the air and contaminate the infusions. He finally concluded that spontaneous generation of microorganisms does not occur at all. Spallanzani's experiment failed to convince Needham, who insisted that the vital force, air, was essential for spontaneous generation and it had been excluded by sealing the flasks. This was answered independently quite later by Franz Schulz and Theodore Swan. Schulz passed air through hot air solution into his flasks of boiled infusions, whereas Swan passed hot air into his flasks. The apparatus used contained a central flask which contained boiled infusions. Air was passed through it from the heated coil. The left flask was a mercury trap through which air escapes after passing through the central flask. In both the cases, the microbial growth did not appear. But the supporters of spontaneous theory were not satisfied. According to them, heat and acid altered the air. The final blow to spontaneous generation theory was dealt by none other than Louis Pasteur. Louis Pasteur was a French chemist who was deeply interested in microbiology. He carried out the famous Swan neck experiment. He took hay infusions in a flask and bent the long neck of the flask into an S shape. This modification allowed air, but not the dust particles and microbes,